Hey everybody. I was feeling a little bit guilty that we didn't get a chance to cover this in class. So if you were wanting more explanation, if you wanted to hear the organelles we talked through, this is for you. If you felt like you totally got it from the reading assignment, then just feel free to ignore this, okay? So I'm going to go through the reading assignment and just going to talk through some of the main ideas, big picture things that you need to know about the organelles, okay? So number one, define organelle. This is going to be a membrane enclosed structure within eukaryotic cells. And now we're going to start our structure and function. So with the cell membrane, the structure here, there's a lipid bilayer, okay? And the whole function is that it's a selective barrier. It allows some things through, but other things not through. And sometimes you'll see it as a cell membrane. Sometimes you'll see it written as a plasma membrane. It doesn't matter. That's a synonym. So, say whatever you want. And then we've got the nucleus. Hopefully you all are aware this contains most of the DNA found in eukaryotic cells. And as it turns out, the nucleus has several parts to it, okay? And so you'll notice, so there's the nuclear envelope. And so this is a double membrane, okay? And this encloses the nucleus, okay? But there are little pores, which you can kind of see. There are little pores that will allow some things in and out, okay? So that's important to know. And also attached to the nucleus here, it's very important to know that the endoplasmic reticulum, specifically the smooth ER, directly connects here to the nucleus, okay? So it's got a double membrane that surrounds it that keeps it separate from the cytoplasm. There's some little pores, and we have that smooth ER that is connected to our nuclear, to the nucleus, like through these different little pores and things, okay? And what else here? What would be coming in and out? Well, RNA might be coming in and out. Different macromolecules might be coming in and out. Proteins might be coming in and out. So that might be going through those pores there. And then the nucleolus, that's this dark patch right here, okay? And that basically, it's like an area of the nucleus, but there's not like a membrane there. And this is where it helps like ribosomes be produced. So this is just kind of like a concentration there, okay? And then there's also chromatin. And chromatin is the name of the DNA and also proteins that make up chromosomes. So hopefully you remember from freshman year, right? Your like chromosome, your DNA model, okay? So there's DNA and proteins in there that make that up, and the technical name for that is chromatin, okay? So that should cover us through the nucleus and all the things associated with the nucleus. So that was letter B there. And then letter C, we've got ribosomes. Now ribosomes are located in two different places, okay? Some of them are free floating in the cytoplasm. They look like little just circles. And some of them are actually attached to the ER. This is the rough ER. Why is it rough? Because there are ribosomes attached to it, okay? And the whole point of a ribosome, the whole thing that it does is that it is going to make proteins, okay? Ribosomes themselves are made up of proteins and rRNA, ribosomal RNA. So they are made of proteins and they make proteins, okay? They're also made of RNA. Ribosomes are not surrounded by like a double membrane type of system here, okay? So the ones that are floating in the cytoplasm, they're more likely to make proteins that the cell itself is going to use, whereas the ribosomes that are attached to the rough ER, making it rough, they're most likely going to produce proteins that the cell is going to excrete, because the rough ER is part of the endomembrane system, which we're going to get into more later, but that's kind of like the post office. So typically those ones are often used to be shipped off elsewhere. Okay, the endomembrane system, here we go. So basically what this is this is our nuclear envelope, which is then connected to the smooth ER, okay? Oh, excuse me, I had that backwards. It's connected to the rough ER, which then connects to the smooth ER, okay? And then From the ER, little vesicles, they're kind of like bubbles, get transported to the Golgi, this green thing over here. The Golgi repackages and sends off vesicles, places. It can send it to other parts of the cell. It can send it to the plasma membrane where then it's bloop, excreted outside the cell. The endomembrane system, its whole function is it's regulating protein traffic. It's helping perform metabolic functions. It's helping package, send things away. It's shipping and receiving. It is all of the comings and goings of the cell are those processes are carried off by this endomembrane system. So it's not one organelle, it's a series of organelles that all work together to kind of be this cell post office, if you will. Okay. Next up, we've got the vesicles. Okay, I want to be really clear with this. For the structure, they are just little bits of the cell membrane that have pinched off and are inside the cell. So it's a double membrane, it's a phospholipid bilayer. And they're just little, basically, membrane bubbles on the inside. And basically, it's kind of like a letter or a package. It's to transfer things through the cell, okay? So if there's something outside that needs to come in, bloop, a little bit of the cell membrane pinches off and goes inside. It does its thing. And then later, if there's things that need to leave, they'll enter a vesicle, and then it'll bloop, fuse with this plasma membrane and leave the cell again, okay? For F, we've got the smooth ER here. And so again, the smooth ER is connected to the rough ER, and the rough ER is connected to the nuclear envelope. The smooth ER does not have any ribosomes. That's what makes it smooth. So what it does is, so the rough ER, part G, we'll just do them together. Rough ER, because there's ribosomes and ribosomes make protein. Rough ER is going to make those secreted proteins. And again, it's connected to the smooth. The smooth, because there's no ribosomes, is not going to make protein, but it's going to make other things. It's going to make lipids. And then it's also involved with a lot of breaking things down. So hydrolysis. So it'd be things like breaking down carbs, detoxifying gross stuff in the cell. You can store ions and calcium. It kind of helps with all these weird metabolic things. It does a lot of things, the smooth ER. Okay, that brings us down to H, the Golgi, which is green right here. The Golgi essentially is going to take products from the ER, the smooth and the rough, and it's going to modify them. So if they weren't quite folded in the right shape yet, if they weren't perfectly ready to go, it's going to get them absolutely ship shape, ready to go. It can store them for a bit, and then when they're ready, they will be sent off, okay, to other destinations, typically through a vacuole, okay, and again, or a vesicle, and those are just little membrane sacs, okay. There was a little subset of the Golgi, and that's the cisternae. So if you notice right here, see how this Golgi is like this series of weird, wonky pancakes? They are not connected together, okay? This rough ER, this is all connected, and the smooth ER, this is all connected. These aren't, but it's like pancake, little spot, pancake, little spot, pancake, little spot. So they're actually, it's like a stack of these Golgi pancakes, okay? And the cisternae is the technical name of these little pancakes, these little sacks, okay? And so in order to get from one sac to the other, things are going to need to travel in like a little vesicle to get from one to the other, to the other, to the other, okay? All right, so now we're down to lysosomes, which down here, they've got it right there. Your lysosome, again, it's a sac of membrane, so it's just your phospholipid bilayer that's pinched off. But the thing is with here, inside are hydrolytic enzymes. What is hydrolysis again? What are we doing? Breaking things down. So hydrolytic enzymes are enzymes used to break things down, used to digest molecules, okay? Oftentimes, lysosomes are a slightly acidic environment because that's what helps facilitate breaking things down. Next, we've got a vacuole. That is not even shown on this picture, but basically a vacuole is a word for a large vesicle. And again, that's just a little bit of cell membrane that's pinched off. And so what happens is vacuoles can be released from the ER and the Golgi. Okay, it's going to contain bigger things. And one thing to note is that the inside of that vacuole is different than what's in the cytoplasm. You can have food vacuoles where that's stored. You can have contractile vacuoles, which like a lot of water critters have. Plants have a large central vacuole. Let's take a look at that for a second. So see this huge central vacuole right here? So plants have a big, huge space in the middle. Animal cells don't have that. But this is where some water storage can happen. Ions can be stored there. In vacuoles, sometimes this is like if you've got waste products, they might get stored in a vacuole, something like that, till they can be released. Next up, we've got the mitochondrion, which is the one that usually people remember. Mitochondrion is single, mitochondria is plural, so don't let that, whatever, like you don't need to know that, but if you see it written either way, that's why. Mitochondria do have a phospholipid bilayer. They're a double membrane, and this is where cellular respiration occurs, which is where ATP is. So usually everyone's like, a powerhouse of the cell, right? Like that's one that everybody remembers. So there we go. One thing to note, there are two like compartments. There's like an inner compartment and like there's a matrix and a little inner membrane space there. We'll learn more about that next unit. So here are the two parts. There's the cristae, which is the inner membrane that's folded. And then there's the matrix, which is the other compartment. So you don't really need to worry about that so much yet, but just know that that is a thing. For letter L, we've got chloroplasts. So that's going to be in a plant cell, and here they are, okay? They are also enclosed by a double membrane, and this is where photosynthesis occurs, right? So this is how plants can convert sunlight into energy and food. Within that, you've got chlorophyll, and that's a green pigment that gives most plants their green color. Not all plants are green. Okay, and inside of here, you can see these little like green coin stacks. Those are called thylakoids. So each little green coin is a thylakoid. A stack is called a granum. And then the stroma is like the goo. It's like the cytoplasm of the chloroplast. It's the goo inside the chloroplast. Okay. One thing to know about here with chloroplasts is they have their own DNA, which is interesting. And so do mitochondria. They also have their own DNA in there as well. Okay, so that brings us to the end of our organelles. And so next we've got a couple little questions to write, you all wrote down. Okay, explain the relationship between the Golgi and the lysosomes and the vesicles. All right. So vesicles carry products from the ER, either rough or smooth, to the Golgi. Okay, so vesicles carry things from the ER to the Golgi. Vesicles also can carry things, products from the Golgi to other places in the cell. A lysosome can meet up with a vesicle and then can digest the food that's in there, or if you put a damaged organelle in a vesicle, the lysosome can meet up with that vesicle and it can break down that damaged organelle. Okay. Number 4, differentiate between the structure and function of vacuoles in plants and animal cells. A vacuole is just a large vesicle, okay? And in plants, we've got that central vacuole that takes up a huge majority. Look at how much of the interior takes up. Massive, right? Lots of area there. And this is going to be a big part of plant growth. It's going to be a big part in plant water regulation. And whereas in animal cells, they're going to be much smaller, and they're just going to be more for transporting things around. Okay, that's not even on this one, but it's just a large vesicle. Okay, number 5. What's part of the endomembrane system? We kind of already talked about this again, but let's go back to it. So we got the nuclear envelope, which is connected to the rough ER, which is connected to the smooth ER. And then the smooth ER can release vesicles. So your next one would be vesicles, which can travel to the Golgi. The Golgi can release vesicles or vacuoles. And then that can end up fusing with the plasma membrane. So if you're keeping count, 1, 2, 3, 4, 5, 6, 7. Nuclear envelope, rough ER, smooth ER, vesicles, Golgi, vacuoles, plasma membrane. All of that is part of the endomembrane system, which again is like your shipping and receiving. Okay. Identify the structure that is not technically an organelle and explain why. That would be ribosomes. Out of this whole list, ribosomes were the only one that were not enclosed in a double membrane. Okay. So there we go. Number 7, which would contain molecules that would aid in hydrolysis of large molecules? Okay, so remember hydrolysis is going to be breaking down. Okay, so which one of our organelles is in charge of breaking down and digestion? That would be the lysosome. Number 8, um, which ones have double membranes? Okay, so that would be chloroplasts and mitochondria. So I want to be clear about something. You can be surrounded by a lipid bilayer, okay? And that's two layers of lipids. But that's separate from a double membrane. A double membrane is literally one membrane and then another membrane. Okay? So mitochondria have two membranes. So imagine like two cell membranes. That's a double membrane, okay? All of the organelles except for ribosomes have a membrane that surrounds them. Because remember, our definition is membrane enclosed structures within eukaryotic cells. That's our definition of organelle. Okay. So our nucleus it's got a membrane. Okay. Vesicles have a membrane. Smoothie R, roughy R, they all have a membrane, but some of them have a double membrane, two membranes. And this would be mitochondria have two membranes, an inner and an outer membrane, and chloroplasts have an inner and an outer membrane. And we're going to learn why that might be notable or interesting or why that might be soon. And then number 9, which ones have their own DNA? That would be chloroplasts and mitochondria. And again, we're going to learn really soon why that may be. Okay, so that's your run through of the organelles. Hopefully this was helpful.